Ebola virus disease, commonly known as Ebola, is a virus caused by the Filoviridae family, a group of lipid-coated virions containing single-stranded negative-sense RNA. This viral family was first seen in 1976 in Germany and Yugoslavia when scientists contracted hemorrhagic fever after handling tissues from green monkeys. Ebola has 5 related species: Zaire Ebola virus, Sudan Ebola virus, Thai forest Ebola virus, Bundibugyo Ebola virus, and Reston Ebola virus. All of these species have been known to cause disease in humans, except Reston Ebola virus, which has only affected primates. The first case of Ebola began in 1976 near the Ebola River, hence the virus's name. In an area known today as the Democratic Republic of Congo, Ebola virus struck the population, killing around 300 people. Only 22% of people who contracted this early virus survived. Other Ebola outbreaks include Gabon, South Sudan, Ivory Coast, Uganda, and Republic of Congo. Ebola was isolated in Africa until the 2014 outbreak. While this outbreak struck West Africa the hardest, it was the first outbreak that spread to other continents. The 2014 outbreak originated in countries such as Guinea, Liberia, Sierra Leone, and Nigeria, and spread to many European countries and the United States. In the US, a total of 10 people were infected. However, only 2 of those people had died by January of 2015. Declared an international health emergency, the CDC and the World Health Organization reported that 11,300 people had died as of January 6, 2015 from Ebola in just Guinea, Liberia, and Sierra Leone. Currently, the World Health Organization states that the case fatality rate of Ebola is 90%. In order to isolate the virus, the CDC implemented airport protocols for travelers. These protocols include testing individuals for Ebola virus as they depart airlines and stalling those who have been in contact with Ebola from boarding planes. Ebola initially begins by entering the body through open wounds and mucous membranes, such as eyes, ears, nose, and mouth. Once inside the body, Ebola attacks the immune system and the vascular system. In the immune system, the VP24 protein found in Ebola attacks the dendritic cells by binding to a transport protein on the surface of immune cells and hinders the cell's ability to terminate viral replication. The attack on the immune system causes T cells to not respond to infections. The bacteria then destroy cells of the adrenal gland that are required to regulate blood pressure by producing steroids. By destroying these cells, the circulatory system in response fails and oxygen levels decrease. Ebola has also been known to damage cells of the stomach lining, causing diarrhea and dehydration. While the host of the virus is unknown, scientists hypothesize that Ebola starts as a spillover infection between humans and either bats or primates such as apes and monkeys. Bats are believed to be natural hosts of the virus. When primates eat these infected bats, they become infected. Furthermore, humans contract the disease through direct contact with these infected animals. The disease then spreads through human-to-human transmission. Contrary to popular belief, Ebola is not an air or waterborne disease. Ebola can only be contracted through direct contact with blood and bodily fluids, contaminated needles and syringes, and infected animals. Research has also shown that some victims have contracted it through sexual contact with the semen of a man who is recovering from Ebola. Other bodily fluids that are infectious agents include vaginal fluids, breast milk, saliva, sweat, vomit, urine, and feces. Symptoms include fever, severe headache, diarrhea, fatigue, abdominal pain, weakness and muscle pain, and unexplained bleeding. Symptoms usually appear within 8 to 10 days after infection. However, signs can appear as early as 2 days and as late as 21 days. After the onset of symptoms, a number of diagnostic tests can be performed to detect the virus, including antigen capture enzyme-linked immunoabsorbent assay, polymerase chain reaction, and electron microscopy. Additionally, those further along in the course of the disease can receive immunoglobulin G and immunoglobulin M antibodies. These antibodies are used to fight viral infections. Those working in healthcare settings, specifically those working with Ebola patients, are most susceptible for contracting the virus. Research has shown that those who were previously infected maintained antibodies of the virus that lasted up to 10 years after recovery. However, it is not known if the antibodies protect against other strains of Ebola virus or if they lead to lifelong immunity. Unfortunately, there is no cure for Ebola. However, through basic treatment measures, patients can significantly increase their chance of survival. These options include intravenous fluids, or IVs, maintaining stable oxygen and blood pressure levels, and treating for other infections if they occur. Currently, researchers are working to produce an FDA-approved drug to cure Ebola victims. There are 2 viable drugs that are being tested. Originally tested by the Public Health Agency of Canada and further tested by the pharmaceutical company Merck, the VSV-EBOV vaccine combined a strain of the Ebola virus and a safer virus. By doing this, researchers hope to train the immune system to defend itself against the virus. Preventative measures include proper hygiene and avoiding contact with infected people, infected animals, and previously infected men's semen. Healthcare workers should take extreme caution when treating Ebola patients. They should wear protective clothing, masks, gloves, and goggles, use proper sterilization methods, and isolate infected patients to minimize risk of exposure.
The hantavirus, also called the hantavirus pulmonary syndrome or HPS, first appeared in the United States in May of 1993. The first outbreak of this illness occurred in an area known as the Four Corners, which encompasses Utah, Colorado, Arizona, and New Mexico, 4 states that meet at their corners. The first recorded case of HPS struck a Navajo man in New Mexico who complained of shortness of breath, which was rare for a being in such physical fitness. Intriguingly, this man's fiancée passed away of the same symptoms just a few days prior. After learning of these strange deaths, doctors were convinced that this couple's story was not solely a coincidence. Therefore, they conducted a wider survey throughout the Four Corners to see if anyone else in the region had died of the same acute respiratory failure. After barely scratching the surface, Dr. Bruce Tempest from the Indian Health Service had already identified 5 other individuals with a similar case. Tests were performed to investigate if this disease was caused by the bubonic plague, but after failing those tests, doctors began to hypothesize that this disease was caused by a virus. Tissues from infected patients were sent to be analyzed by virologists who discovered the pulmonary syndrome was related to an unknown hantavirus. Researchers were aware that other common types of hantaviruses were transmitted by rodents. As a result, they narrowed their search to trapping any kind of species of rodent that appeared in the Four Corners region, especially those found in and near the homes and workplaces of those who passed away from this illness. After trapping and examining nearly 1,700 different rodents, the Peromyscus maniculatus, or the deer mouse, was established as being the primary host of the hantavirus. Studies showed that about 30% of the deer mice that were trapped tested positive for the infection of the hantavirus. Other rodents also carried the disease, but the occurrence in those animals was not quite as common. After conducting a case control investigation in which a household of those infected was compared to the control household, or one that had not been exposed to the virus, the hantavirus was isolated. In November of 1993, the Special Pathogens Branch at the CDC was able to grow the virus in the lab, and the U.S. Army Medical Research Institute of Infectious Diseases followed shortly after. The hantavirus pulmonary syndrome, or HPS, was originally referred to as the Muerto Canyon virus and later the Sin Nombre virus. Further studies on the tissues belonging to the individuals who had already passed of this respiratory distress proved that the hantavirus actually existed before the first recorded outbreak. In fact, the first case was seen in 1959 in a man in Utah. The virus itself had simply gone undetected. However, the question still remained as to why the hantavirus specifically struck the Four Corners region. Before 1993, the area had been barren. A drought swept the region, and with that drought came a lack of vegetation. In the beginning of 1993, the Four Corners experienced lots of rain and snow. Consequently, there is lots of water to revive this arid land, and the deer mice had plenty to eat to keep their numbers strong. To this day, no case has ever been reported in the U.S. that involved person-to-person transmission of the disease, like one would expect in an infection like the common cold. However, researchers have uncovered that HPS can be caused by a multitude of hantaviruses. There is not just one. For example, a 33-year-old man from Florida who had never even visited the Four Corners was diagnosed with this virus in 1993. Similarly, another investigation found that the Black Creek Canal virus, which is another hantavirus that is carried by the cotton rat, or Sigmodoum hispidus. In November of 2012, the National Park Service announced that 10 cases of HPS were found in people who had visited Yosemite National Park. However, the majority of people infected are exposed to the virus in their very own homes. Other cases have also appeared in Argentina, Brazil, Paraguay, Chile, Uruguay, and Canada, confirming this disease is widespread. While the hantavirus has been found in a myriad of rodents, such as the white-footed mouse in New York, evidence proves that most cases are linked to the Sin Nombre virus, which is carried by the deer mouse. Furthermore, the majority of the cases are located in areas conducive to the rat's life. Typical areas would include barns, sheds, and outhouses. In those locations, the rodents leave the virus via urination, secretion, and salivation. Therefore, people obtain the virus simply by breathing in the contaminated air, a process known as airborne transmission. Other pathways of transmission can include direct contact, when a rodent carrying the virus bites someone. This is the most uncommon transmission strategy. It is also believed that people who touch something that has been contaminated by the virus and then touch their noses or mouths can also become infected. Eating food that has been exposed to the virus can cause the virus in a human as well. Because the virus is airborne, it is important to remember to not sweep, vacuum, or do anything that would stir up dust when in an area that could potentially be infected by the rodent. It has been estimated that symptoms can appear between 1 to 5 weeks after exposure. Early symptoms can be detected by experiencing fever, muscle aches, and fatigue. Dizziness, headaches, abdominal pain, nausea, diarrhea, and vomiting are also attributed to the hantavirus. After about a week following exposure to the urine, droppings, or saliva of the infected rodent, those who have the virus might endure coughing and shortness of breath. Your lungs will become filled with fluid. HPS is very serious. About 38% of the individuals infected have died as a result. Because the early symptoms of HPS are so ambiguous, it is seemingly impossible to identify the virus early on. However, if fever and fatigue are associated with a shortness of breath, especially in a rural area where the rodents are prone to reside, the patient should immediately consult a physician. An exam could reveal kidney failure, low blood pressure, low oxygen levels resulting in blue coloration of the skin, or acute respiratory distress syndrome. Although there is no sure cure or vaccine to treat the virus, individuals who have become infected can improve their health status with oxygen therapy in an intensive care unit. Other treatments could include breathing tubes or machines, a prescription of ribavirin, a medicine used to treat kidney problems, or using machines to oxygenate the blood. In order to lessen the chances of becoming infected by the hantavirus, one should set up traps around his home. Storing or throwing away open food is another easy solution that could potentially save a life. Hantavirus pulmonary syndrome had been diagnosed in 637 people until December 31, 2013. 606 of those cases were identified after 1993, and 36% of all reported cases have been terminal. More males have acquired this disease than females. Men have accounted for about 63% of individuals with HPS, while women make up 37% of the infected population. Furthermore, there is not one specific age in which HPS is most prevalent. Ages of those affected range from 8 years to 83 years, the average person being 37 years old. While this virus can impact anyone, it is seen most regularly in white people, who accounted for 78% of all cases. 18% of all cases have occurred in American Indians, 2% in African Americans, and only 1% in Asians. Cases of HPS have been reported in 34 states. However, more than 95% of those cases appeared in lands west of the Mississippi River.
Chasing down Ebola as reports multiply about possible cases showing up in this country.
Breaking news on the Ebola outbreak, the CDC declaring a level 1 activation of its emergency operation.
Suspected case of deadly hantavirus in La Plata County.
More public concern about hantavirus.
The hantavirus has killed 3 and has threatened the lives of thousands in Yosemite National Park.